Good morning, Professor and classmate. I'm Subin Bae in Protein Research Lab. Today, I'm introduce the proteomics and talk about what I learned in the lab. Proteomics is the large-scale study of proteomes and the analysis of the protein complement to the genome. Proteome is a set of protein produced in an organism, system, or biological context. While the genome is a later constant entity, the proteomes differ from cell to cell and is constantly changing through its biochemical interaction with the genome and the environment. Using the more inclusive definition of proteomics, many different areas of study are now grouped under the proteomics. These include protein expression profiling, post-translational modification, protein-protein interaction, structural proteomics, functional proteomics, proteomimicry. Protein post-translational modification increase the functional diversity of the proteome by the covalent addition of functional groups or proteins, proteolytic cleavage of regulatory subunit, or degradation of entire proteins. Changes at the transcriptional and mRNA levels increase the size of the transcriptome relative to the genome, and the myriad of different post-translational modifications exponentially increase the complexity of the proteome relative to both the transcriptome and genome. PTMs are chemical modifications that play a key role in functional proteomics because they regulate activity, localization, and interaction with other cellular molecules such as protein, nucleic acid, lipid, and cofactor. These modifications include phosphorylation, glycosylation, ubiquitination, nitrogylation, methylation, acetylation, lipidation, and proteolysis, and influence almost all aspects of normal cell biology and pathogenesis. Protein theory play a crucial role in redox signaling, in the regulation of enzymatic activity and protein function, and in maintaining redox homeostasis in living systems. The reversible PTMs in particular are major components of redox signaling and are involved in the regulation of various cellular processes under physiological and pathological conditions. Increase in salinity levels increase ion toxicity, primarily due to the accumulation of NaCl, restriction of mineral nutrient uptake, loss of osmotic potential and ROS production, and alteration in various physiological processes. The increased ROS production causes damage to cells, but in addition they induce signals for enhancing some protective mechanisms to scavenge ROS, as well as damaging agents contributing to third injury in plants. The scavenging of ROS to be essential for tolerant to salinity. Next, I will describe the experimental method used in my research. MBT depth and advanced blue staining for the detection of H2 and cell death. Depth and MBT staining method can be the rapid method to characterize ROS accumulation in plant cell under the abiotic stresses. Tissue is based on the ability of cells to reduce MBT. MBT especially react with oxide and form a purple-blue formation precipitate. Advanced blue dye have been used as a viability assay on the basis of its penetration into non-viable cells. Also, the method is subject to error because it assumes that damaged or otherwise altered cells are not capable of repair and therefore are not viable. 2 weeks old Arabidopsis was attacked stressed by vacuum infiltration, and the whole plant were immersed and stained in the solution. Co-IP is a powerful technique that is used regularly by molecular biologists to analyze protein-protein interaction. Prepare lysate from cells or tissue samples which express the target protein is the first step of the co-IP. In order to preserve the intact of protein-protein interaction, the lysis purpose should use non-ionic detergent. Then, the target protein are captured by specific antibodies from total lysate. The resultant immunocomplex can be precipitated using a resin that is conjugated with IgG binding protein Ag. The third step is washing. Irrelevant non-binding protein agent and any protein that are bound are eluted by a series of washes. Then, the bound proteins which eluted are analyzed by SDS phase and immunoblotting and mass spectrometry. Bimolecular fluorescence complementation, also known as BIFC, is a technology typically used to validate protein interactions. It is based on the association of fluorescent protein fragments that are associated to components of the same micromolecular complex. Proteins that are postulated to interact are fused to unfolded complementary fragments of a fluorescent reporter protein and expressed in live cell. Interaction of these proteins will bring the fluorescence fragment within proximity, allowing the reporter protein to reform in its naive three-dimensional structure and emit its fluorescent signal. Thank you for listening to me.